Hello everyone. The objective of this lecture is to understand the central dogma of biology, which is DNA to RNA to protein. The central dogma of biology provides the basic framework for how genetic information flows from a DNA sequence to a protein product that's made inside the cells. This process of genetic information flows mainly in two steps: transcription and translation, which we will be discussing in this PowerPoint. So the next question is, if DNA cannot leave the nucleus, how can it get the instructions out to make the proteins needed to survive? So the answer is this molecule, which is called RNA. RNA stands for, as you all know, ribonucleic acid. It contains the sugar ribose, and it's single-stranded, and it contains 4 nitrogenous bases, just like DNA, but instead of thymine, it has uracil. So let's look at the two structures side by side. We have the structure of DNA and RNA, and as you can tell, RNA is single-stranded as compared to DNA. Now, the three main differences between DNA and RNA that I just mentioned is they have two different sugars. DNA has deoxyribose, RNA has ribose, and there's a difference of nitrogenous base between the two. DNA has thymine and RNA has uracil, and it's DNA as double-stranded and RNA is single-stranded. Now let's talk about the three main types of RNA. The first one is messenger RNA. Messenger RNA is present in the nucleus, and this is the RNA that carries the message from the gene on the DNA strand outside of the nucleus. So in other words, the entire genetic information is transcribed from DNA to RNA. These are the complementary strands, and you can see for A, there is a complementary uracil instead of thymine. So the second type of RNA is a ribosomal RNA. Ribosomal RNA is present in the cytoplasm, and this is where the proteins are assembled. And each ribosomal RNA is made up of 4 subunits of ribosomal RNA. Each ribosome is made up of 4 subunits of ribosomal RNA and 82 proteins. So that makes up 1 ribosome. The last of the 3 types is the tRNA, and the name comes from the structure that looks like T and also its function, that is to transfer. It transfers amino acids from the cytoplasm and brings them to the ribosome, where these amino acids will be complementary to the codons on RNA, and more about this on mRNA, and more about this when we discuss in detail transcription and translation. So what's a protein? Protein is a chain of amino acids. It's one of the 4 macromolecules, and this is the structure of primary protein, where you see amino acids are attached to each other in a single strand. The 2 major steps of protein synthesis are transcription and translation. In transcription, all the information from DNA is transcribed over to mRNA in the nucleus of the cell. In translation, this messenger RNA goes and attaches to the ribosomes, and tRNA brings in amino acids, making the protein. So let's look at the steps of transcription. The first thing is DNA helicase will break the helix, and 1 strand of DNA will get transcribed. And this strand starts with the 3 nucleotides TAC, TAC. Okay. Now, RNA polymerase comes and attaches at the beginning, and it starts transcribing the information from DNA over to RNA. And the example is here. If this is the segment of DNA, then this is the complementary segment that has been transcribed on mRNA. And then mRNA can leave the nucleus. But before it leaves, all the information has to be edited. Now, not all the codons on mRNA are transcribable. They cannot be coded. So there are segments of DNA, there are segments of RNA that will be taken out. They are excised, and this is called the junk, or the introns. They will stay in the nucleus, whereas the exons, which is the DNA that will be coded into protein, exits. Now there is protein called spliceosome, which does this function. And before this mRNA can leave the nucleus, there is a long poly A tail that's attached to one end of the mRNA and a 5' cap on the other end of mRNA. So this is to prevent mRNA from degrading the enzymes that are present in the cytoplasm that can actually degrade the mRNA. Now these exons splice together in different sequences to produce different kinds of proteins. So fun fact, over 98% of the human genome is non-coding. And this is probably due to evolution. And as a result, only there are 25,000 genes that actually produce 100,000 different proteins. Okay, so now let's look at what we have learned so far. We have looked at DNA that needs to be transcribed over to mRNA through the process of transcription. And this mRNA is now going to leave this nucleus, come into the cytoplasm, where it will attach to the ribosome. tRNA is going to have complementary nucleotides to mRNA, and to each of these codons is an amino acid attached. Now please go over this transcription animation. Just for better visual and better understanding, I've given you different links, so it's your choice to go through all 3 of them, or you pick and choose which one you would like to view. So moving on to step 2, which is translation. In translation, it's basically coding. So now the mRNA, every 3 nitrogen bases become as one word, and we call it as a codon. So amino acids are abbreviated in 3 letters, and each amino acid is complementary to a codon. For example, for methionine, AUG, that means if the mRNA has nitrogenous base in the sequence of adenine, uracil, and guanine, then methionine will come and attach to these codons. So let's look at our codon chart. Now, how to read this codon chart? There are amino acids. There are only 20 amino acids that make up all the proteins. So if you look on the first position, all 4 nitrogenous bases are listed. On the top, second position, and on the right side, you have the third position. Now, if you have to find the corresponding amino acid to U, you will look at the first position U, second position U, and go across on the third position U, and this will correspond to your amino acid phenylalanine. And if you realize the third base can be changed. So third base, it can be U, C, A, or G, and it's going to code for different amino acids. And this is called the wobble effect. So in order to practice, I would like you to try these, GCU, and see what it corresponds to. I have given you the answers, and see if you can get the same answers. Now, let's get into the details of translation. So translating of an mRNA codon into a protein takes place with the help of tRNA. So tRNA has amino acids attached to the other end. They come along, and the first amino acid is always methionine. For every single protein, the first amino acid will be methionine, and the last would be the stop codon, indicating that's the end of the gene, and this is the length of the protein that you're going to get, or a polypeptide chain. So AUG is the beginning of every single mRNA and ends with stop codon. Therefore, first amino acid as methionine and the last as the stop codon. Now, mRNA attaches to the ribosome, and this gives you the steps. Each codon of mRNA molecule will move through the ribosome like it's moving through a conveyor belt. The tRNA will bring the proper amino acid to the ribosome. The codons, or actually the nucleotides, or the nitrogenous bases on tRNA are called anticodon, and the nitrogenous bases on mRNA are called codon. And your codon chart is read according to mRNA. Amino acids bond together with the help of peptide bonds, and this amino acid chain is called polypeptide. This polypeptide chain will then eventually become a protein. And how it becomes protein is by having different orientations. It can be a primary protein, secondary protein, tertiary protein, or quaternary protein. Now, for the primary protein, there's only one single polypeptide chain, which can be straight, which can be helical, or which can be spiral. So these are the different shapes that polypeptide chain will take, and then eventually it's called the protein. For secondary protein, there will be more than two polypeptide chains, and these two chains can either intertwine with each other, become a helix, or they can spiral around, or they can take any other shape. So likewise, the tertiary and the quaternary proteins become more and more complex. Now we will talk about this in another lecture, but I just wanted to give you an overview and give you the difference between the polypeptide and protein. So polypeptide is like raw protein, and then when you give a specific shape to polypeptide chain, it becomes a protein. So we've already talked about the start codons and the stop codons. That's at the beginning and the end of the polypeptide chain. So there are three stop codons: UAA, UAG, and UGA. Now here are more translation animations this time to give you an idea and also visuals for what I've been describing. So hopefully you will stop at that slide and go through all the animations and get a better understanding of this process. This is an overview of translation that I just talked about. Please pause at this slide and kind of go through it and understand what's going on. And finally, this is the process or the central dogma of biology, which is DNA to protein. That's what we discussed in this lecture, and hopefully you understood it. If you have any questions, as always, you can ask me in the classroom, or you can email me and I'll be happy to answer those questions for you. Thank you.